All right, now we're moving on to chapter 13.3, and we're going to be talking about meiosis. But before we get into that, I want to go over just some brief differences between mitosis and meiosis. So one thing that we can say is that mitosis comes from one parent or one cell, and then it can divide into two cells. And the offspring, which are these two cells, are going to be identical to the parents. You can almost say they're like clones because they're ending up with the same number of chromosomes as the parents, and they're going to look identical to the parents. So their genes are going to look exactly like the parents' genes. But in meiosis, first of all, there's two steps to meiosis. There's meiosis 1, and then there's meiosis 2. When we go through these in the next couple minutes, you're going to kind of see the difference between these two and how meiosis 2 actually looks a lot like mitosis, and meiosis 1 is a little bit funky. But as we go through meiosis, you're starting with a cell that's going to divide into two cells, and that cell is going to divide into 4. So you're going to end up with 4 offspring cells that are going to come out of meiosis rather than 2 that are going to come out of mitosis. Another important thing to know is that these 4 cells that come out of meiosis, they're not going to be identical to the parent. All right, so they're going to look really different, and we're gonna, not really different, but you can tell that they're related, but like siblings, but they're not going to look like twins. They're not going to look like these clones over here. Okay. And then we're going to talk about why this happens. Why do siblings, even though they're related, not look identical? Or why do offspring, though they're from the parents, not look exactly like the parents? So like I said, you're going to have 4 daughter cells rather than 2. 2 comes from mitosis, and then you're going to get 4 in meiosis. And when you get these 4 cells, they're only going to have half as many chromosomes as the parent. All right, so you're going to literally go from diploid, which means you're going to have 46 chromosomes, to haploid, which means you're going to have 23. Which is not like mitosis. You're going to end up with 46 eventually, okay? All right, so let's start talking about what happens in 13.3. So when we talk about meiosis, we said that meiosis occurs in the sperm and the egg before they even get fertilized, all right? So this process that we're going to kind of go in depth with is happening both in the sperm and the egg before they even meet up. The cool thing is, the sperm will go through the whole process of meiosis before fertilization, but the egg actually stops at a phase of metaphase before it gets fertilized, and then after it gets fertilized, it will continue to finish the rest of meiosis. So that's pretty interesting for the egg to go through. All right, kind of backtracking a couple chapters, we talked about how the interphase is where the interphase is where DNA replication occurs, right? So DNA replication means that they are being duplicated and so on and so forth. So now that we talked about interphase occurring before mitosis and meiosis for both of them, and that's when DNA replication occurs, let's talk about something specific to meiosis. So I mentioned up here that meiosis actually has 2 stages. It has meiosis 1 and meiosis 2. Unlike mitosis, which all just occurs in one stage. So as we go through talking about meiosis, we're going to talk about each of the stages and then what happens in them. And then you can get a better understanding of why that is so important for our gametes to go through them. So to start off, meiosis 1 has 4 phases. So if you remember the kind of acronym that I used where we said it was something called PMAT, you can also use the same thing when we talk about meiosis 1 and meiosis 2, but we'll talk about what you can use to differentiate them between the first phase and the second phase. So meiosis 1 has 4 phases, and these are the phases. You have prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. Yeah, they're all the same from mitosis, except for the 1. And you'll also notice in meiosis 2, they're all going to be the same phases, except they're just going to have a 2 next to them. So it's not going to be really hard to remember what the name of the phases are. The name of the phases will stay the same. It's just the numbers are going to differentiate which phase you're going to be in. All right, so let's talk about what happens right before PMET. I know I said that we have interphase, which is where DNA replication occurs, but let's look at what happens in meiosis during interphase. So in interphase, what's happening is that you have chromosomes. You're going to have chromosomes from mom and dad, mom, dad, and they're going to have 46 chromosomes in them. So if you remember, this means that they're diploid, okay? It's because there's 46 chromosomes in them. So in interphase, you're going to have these single chromosomes. They're going to go through DNA replication. So they're going to make a copy of each so that now you're going to have 2 of them. Then we move on to prophase. Prophase, it gets a little interesting. So just bear with me as I talk about this. So in prophase, you're going to have, take one of these cells, right? So you're going to take this cell that has been duplicated for its chromosomes, and they're going to come together and they're going to make a homologous chromosome right here. So remember that we talked about homologous chromosome means that they're identical, right? So that means they're the same size, they're going to have pretty much the same kind of structure and shape. So you have homologous chromosomes right here. You have some from mom and some from dad. And then you have some homologous chromosomes here, some from mom, some from dad. Notice I try to kind of vary them, that these are a little bit longer than these. So they pair up with the ones that are very closest to them. All right. So obviously I said the pink is going to be mom, or the reddish color, the blue is going to be dad. And then when they're when the mom pairs up with the dad, they're going to pair up according to similarities in structure and shape, and that's going to make your homologous chromosome. Now, in the homologous chromosomes, you have something called cohesions. This is going to help hold the sister chromatids together. In mitosis, we also talked about cohesions, which are kind of like proteins that kind of line up along the side of the chromosome, and they're going to help hold these chromosomes together to make the pair, the sister chromatids. Also, you have something called synaptonomal complex, and which that basically means that they're holding all 4 of these together. So it's kind of like the mom and the dad are being held together by this complex. Now let's move on. So at the end of prophase 1, what you're having is something very, very interesting that is very unique to meiosis, and meiosis 1 in particular, and even more in depth to prophase 1. So in meiosis 1, in prophase 1, you have something called crossing over. So what happens is, we looked at these chromosomes over here that make these homologous chromosome pairs. They're going to do something funky where the pairs that are closest to each other, so you can see that the blue pair that's closest to this red one here, the blue chromosome that's closest to this red chromosome, they're going to do this thing called crossing over. So they're going to cross over, and if you kind of can look at it, if I zoom it in, one chromosome from the mom and one chromosome from the dad are kind of going to link up, and when they link up, they're going to link up at a spot called the chiasmata. All right, so that's where the crossing over is happening. When they cross over at the chiasmata, they're going to do something. And this is what they're going to do. So during the crossover, they're going to swap sections of the DNA. So mom is going to swap sections of DNA with dad. Dad's going to swap sections of DNA with the mom. And this is called recombination. Now, as they swap sections, they're going to do it at the same location, all right? This is important so that they're both going to have the same number of genes. If they don't do it at the same location, what if the mom chromosome ends up with 1 or 2 extra pairs of genes than she originally had, or vice versa with the dad? That would not be a good thing. So when they do recombination or crossing over, they're going to do it at the same location, and they're going to make sure that they're switching kind of at the same genes. So they're all going to have the same number of genes. All right. Now, this recombination is super important. All right. The reason it's super important is because it's creating variety. Why is it creating variety? This is what is causing siblings to look different, even though they're related. All right. We talked about in mitosis, they all are identical. But in meiosis, that is not the case. In meiosis, they look different. Parents look different from their kids. Kids look different from their siblings because of this crossing over. All right. So it's going to create a different kind of variety. But twins, they actually look identical if they're identical twins, not fraternal. They look identical because they're coming from the same sperm and their same egg. So they don't have as much variety in their genes as they would just regular siblings that come from different sperm or different egg, okay? Because when you have twins, you're going to be implanted the same way. All right, so that is what I'm talking about when I talk about crossover. If you need to watch this again, do it. Basically, all you need to remember is crossing over happens in meiosis 1, in prophase 1. You have mom and dad going to be kind of getting a little intimate right here, crossing over some genes at the location where they cross over is called the chiasmata. Because they're swapping genes in the DNA, it's called recombination, which gives them variety so that siblings look different, parents look different from their kids, so on and so forth. So now that we just finished talking about prophase 1 and interphase, so interphase and then prophase, now we're going to go into what happens next, which is metaphase. In metaphase 1, what's going to happen is that, just like we talked about in metaphase and mitosis, our homologous pairs, or your homologs, are going to line up at the metaphase plate, all right? So once they line up at the metaphase plate, your microtubules are going to come out and attach. Microtubules should already start coming out in prophase. I didn't draw it over here, but if I should, give me a second, you're already going to start seeing some microtubules coming out, all right? Just like in mitosis. So in metaphase, microtubules are going to attach to these chromosomes, the homologs, and they're actually attached to each other. I just didn't do a really good job of drawing them together. But once they line up at the metaphase plate, what they're going to do is the microtubules are going to attach to the kinetochores on the homolog. So remember how I said centromeres, we talked about centromeres and kinetochores. Centromeres hold the sister chromatids together. So the centromeres would be between these two and would be between these two. But the kinetochores are going to be on the outside. Okay, they're going to be on the outside, and that's where the microtubules are going to attach to. They're going to attach to the outside part, all right? So this is going to help them in the next phase pull them apart. So let's talk about what happens in the next phase. So in anaphase, which comes after metaphase, we talked about PMAT, so prophase, metaphase. Now we're up to anaphase. So let's see what happens in anaphase. So in anaphase, we are pulling apart the homologs of the chromosome. So the homologs are going to separate because of the spindles that are attached to the kinetochores. And the spindles are going to pull the chromosomes to each side of the pole. So you can see how the spindles are pulling them to each side of the pole. And the sister chromatids, though, they're going to be attached to each other. So again, I did not do a very good job, but just imagine that these are attached. These sister chromatids are attached to each other by something called the centromere. So they're attached by the centromere and they're getting pulled apart. So remember, sister chromatids, which are these two chromosomes together, are being pulled apart so that they're no longer homologous chromosomes, because the homologous chromosomes were when they were 4 of them together. All right? So that's what's happening in anaphase. Anaphase, the microtubules are pulling apart the homologs so that they're now only sister chromatids, and they're moving them to opposite sides of the pole. So let's talk about what happens in telophase 1 and cytokinesis. These kind of happen pretty much at the same time. So, well, not same time, but they're happening simultaneously. So it's not like it's telophase and then pause, cytokinesis. They're kind of just happening together. So what happens is in telophase 1, just like we talked about in mitosis of telophase, these two cells are going to pinch off from each other. But the interesting thing is that they're both going to have sister chromatids inside of them, all right? So as they're pinching off, they're going to end up looking like this, and they're both going to have some mom and dad DNA, some mom, dad DNA, some mom, dad DNA, some mom, dad DNA. So if you can see, there's some blue and red in here, blue and red in here, blue and red in here, blue and red in here. So there's variety happening as they go from telophase to cytokinesis, all right? So what exactly is happening? In telophase, you're going to start with each half of the cell is going to be haploid, all right? So each half of the cell is still going to have 2 sister chromatids, but they're going to be haploid. So they're only going to have half of what they started off with, which means that they're only going to have 23 chromosomes in each. In cytokinesis, this is when they kind of pinch off. So now you have 2 haploid daughter cells, all right? So each of them are going to have 23 chromosomes. And then we're going to see what happens after that in meiosis 2. Another thing to remember is we talked about this in mitosis, all right? In animal cells, when cytokinesis is occurring, they're going to create something called a cleavage furrow. So if you can see, the cleavage furrow is starting right here, and as they pinch off, they're going to end up with having plasma membrane surrounding the cells. But in plant cells, you don't have cleavage furrow. They don't have plasma membrane like animal cells do. They have something called a cell wall or cell plate that's going to form. So that's going to form right between them. Instead of plasma membrane, it's going to be a cell plate, and that's going to distinguish it from a plant cell to an animal cell. Now, as we transition from meiosis 1 to meiosis 2, remember, there's no process in between where these chromosomes are going to go through duplication. So how, like we said in interphase only occurs right before meiosis 1. It does not occur again before meiosis 2. So make sure you remember that. No chromosome duplication occurs between meiosis 1 and meiosis 2. All right, so now let's move on to meiosis. All right, so let's talk about what happens in meiosis 2. So meiosis 2 also has 4 phases like meiosis 1. It has prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So they're very similar to meiosis 1, except for the part that they have 2. Now, the interesting thing about meiosis is that's going to be very similar to mitosis. So you're going to see how meiosis 2 is a lot more similar to mitosis than meiosis 1 was. Because in meiosis 1, we had crossing over that mitosis does not have. And that's because in mitosis, the daughter cells they're exactly identical to the parents. But because of them crossing over in meiosis 1, that's why we get offspring that are different from the parents and from the siblings, unless they're identical twins. All right, so let's see what happens in prophase 2. So in prophase 2, you have your sister chromatids. You have the formation of spindles, sister chromatids, formation of spindles right here. But the thing that you need to notice is that in prophase 2, we're starting off with 2 cells. We're not starting off with 1 cell like we did in meiosis 1 or in mitosis. We're starting off with 2 cells because that's what we ended with in meiosis 1. All right, so like I said, in prophase 2, your spindles are forming, and at the end of prophase 2, they're going to start to line up at the metaphase plate so that they can get ready for metaphase. So in metaphase 2, this is what's going to happen. They're going to be lining up at the metaphase plate. I did excuse my drawing, but they are still going to be attached, okay? So just imagine that they're still attached by their centromeres. I did an awful job of drawing them very far apart, but they're going to be still attached to each other by their centromeres. So the sister chromatids are at the plate now. Because of crossing over in meiosis 1, the 2 sister chromatids are not identical, all right? They are not identical. That means they have some mom and some dad in this cell, some mom and some dad in this cell. So if you can kind of see, I kind of colored them 2 different colors here. Blue and red, blue and red. These chromosomes that are just the same color, those were the ones that were the outside of the homolog pair, so they did not do crossing over. It's only the 2 chromosomes that are on the inside that they cross over. And then kinetochores are going to attach to the microtubules. So like I said before, remember that centromeres are connecting the sister chromatids together. And then the kinetochores are going to be on the outside that the microtubules connect to onto the sister chromatids. So that's what they're kind of going to look like. These are the kinetochores. And then we are going to move on to anaphase and see what happens in anaphase. All right, so in anaphase, what's going to happen is you're going to have 2 cells like we started off with, and they're going to be pulled apart. So as you can see, the microtubules are pulling them apart, the centromeres break, and they start to move to opposite sides of the poles, all right? Then in telophase 2 and cytokinesis, we are going to see, sorry if my hand's super shaky, we're going to see the final product, okay? So we're going to see the two cells that are starting to form their cleavage furrow. So each chromosomes are now at the opposite cell. So you have a little bit of, you have chromosomes that are in both of the halves. The nuclei is going to start to form, and chromosomes are going to start to decondense. And then they're going to pinch off, and then you're going to have cytokinesis, which is the process of pinching off. Remember, in animal cells, it's going to be plasma membrane. They're going to form the cleavage furrow. And then in plant cells, you're going to have cell plate or cell wall. All right? So cytokinesis is when the cytoplasm is going to divide. You're going to have organelles separating and so on and so forth. And then you are going to now end up with 4 products. You have 1, 2, 3, 4. All right. So you're going to end with 4 different sex cells. All right. They're going to be genetically unidentical. They're not going to look the same. All right. And each of them now are going to have 23 chromosomes. And that's what makes the gametes haploid, all right? So the gametes are going to be haploid because they're going to have 23 chromosomes, and that's what we're going to end up with at the end of meiosis. So now let's look at some reminders, all right? So 3 special events that occur in meiosis that are different from mitosis is 1, you have crossing over, which is also called synapsis, all right? So this occurs in prophase 1 in meiosis 1, and this is when the homologs, which are closest together, are going to exchange genetic information. So if I kind of zoom in a little bit, if I have the mom here, so if I draw 2 moms, right, and then I have, let me get a blue pen really quick. If I have dad, all right, so let's color dad blue. And let's color mom pink, all right? Now, at crossing over, what is happening is that these 2 chromosomes right next to each other, they're going to switch genetic material, right? So what's going to happen is the chromosomes that are on the end, they're going to stay the same way because they're not part of the sharing. But the chromosomes that are in the middle, they're going to be crossing over, all right? And they're going to be sharing some information. So what ends up happening then is that you're going to have this chromosome stays the same, but then this chromosome is a little pink, but it has now some blue to it. This chromosome is going to be mainly blue, but it's going to have some pink to it, all right? And then this one is going to stay blue. All right. So that is what crossing over looks like. So now you have a homologous pair, but they have some DNA that they have shared. All right. What's another thing that's special about meiosis? Meiosis, they have homologous pairs at the metaphase plate. So in mitosis, we only have sister chromatids. Here we have homologous pairs, some with, so they all have some have mom, some have dad, so on and so forth. They're also going to separate as by homologous in anaphase 1. So that means after metaphase 1, in anaphase 1, these homologous pairs are going to separate, so they're going to end up as sister chromatids. In mitosis, we have cohesions that are cleaved at the end of metaphase. So these cohesions are located between the chromatids to help them stay together. But in meiosis, they're going to be cleaved at the end of anaphase 1 when the homologs separate. And then the centromeres, which are between the sister chromatids, are going to be separated in anaphase 2. Because remember, in meiosis 1, you're separating the homologous pairs. And then in meiosis 2, you're going to be separating just the sister chromatids, all right? So if you need to rewatch this video and go back to the rewatching what meiosis 1 looks like or meiosis 2, that could be helpful for you guys, all right? So this is the end of chapter 13.3. I will be putting up more assignments for you guys to do, some worksheets and stuff. If you have any questions, please feel free to email me. And please do rewatch these videos. I'll see if I can find some other videos that kind of show you a little bit clearer what's happening, and then we'll go from there.